Hello and welcome. I'm Blake Varenholtz, and this video is being made for Chem 855, Principles of Biochemistry, as part of my Master of Science in Education and STEM Education program at the University of Nebraska at Kearney. Shout out and thanks to Dr. Frank Kovacs for a great class this summer. I've learned so much in such a short amount of time. My chosen topic today is protein synthesis, so let's jump right in. As a general overview, this presentation covers the central dogma of biology. Really, it's just that DNA codes for RNA, which codes for proteins. We'll get a little bit more specific as we go along the way. Let's start by looking at the structure of deoxyribonucleic acid, or DNA. This is the molecule that carries genetic information that determines the phenotype of an organism. It has two linked strands that wind around each other in a way that looks like a twisted ladder, otherwise known as a double helix. You'll notice that this image labels the major groove and the minor groove as part of the shape. The backbone of each strand is made of an alternating series of deoxyribose sugar and the phosphate group linkage, and each unit is attached to one of four base pairs, which we see here labeled as A, T, G, and C. Let's take a closer look at each of those. You'll notice here that I have it listed as attached as a 5' and 3' direction. We'll get more into that in the next slide as well. Here we have the structure of our four nucleotides. Each has three components. The first two are the same on all four, but the third sets each apart from the others. Part 1 is a pentose sugar, which is deoxyribose for DNA. Let's take a look at the carbons on these and double check. If we count each point here, we should got 1, 2, 3, 4, 5 sugars, and we should find those 5 sugars on each of these sugar rings here. On the last slide, I noted the 5' to 3' linkage, and this is what is meant by that. The fifth carbon, 1, 2, 3, 4, 5, the fifth carbon here is linked to the third carbon of the next one, 1, 2, 3, and the same going in the opposite direction on this side, 1, 2, 3, 4, 5, down to 1, 2, 3 on this side. And those are all linked to our next common part, which is the phosphate group. So we'll see the phosphate group is what links each of these, linking the sugar on each side there. And finally, we have 4 different nitrogenous bases: thymine, adenine, guanine, and cytosine. These all bond to each other in pairs through what's called Watson-Crick pairing. Notice adenine and thymine form 2 hydrogen bonds, and then cytosine and guanine form 3, most of the time at least. A change in either of these pairings would cause a mutation, or could cause a mutation, so it's important that they are paired correctly, and that's why it's important that we have the 2 linkages on these ones and 3 on these ones. This also means that each strand reads complementarily to the other. So we read one in the 5' to 3' direction and the other in the 3' to 5' direction. That leads us to our next slide. Excuse me. DNA transcription. An enzyme called DNA helicase unzips the base pairs that allows for access to individual strands. Notice the top strand goes from the 5' to 3' direction, and it's not used for transcription. The bottom strand goes from 3' to 5' and is used for transcription. It's read by an enzyme named RNA polymerase. Sorry, before this was not showing DNA helicase. That's somewhere up here, unzipping it, the DNA up here. In the initiation phase, RNA polymerase locates the protomer that marks the beginning of the appropriate gene, and it attaches to the strand of the DNA and begins to build an mRNA. We'll discuss more on that structure in a moment. Phase 2 is called elongation. RNA polymerase moves along the DNA strand and collects and attaches free-floating surrounding nucleotides in a 5' to 3' mRNA strand. Termination is the final step for RNA polymerase as it releases the DNA strand and also the mRNA strand. Let's zoom in onto RNA for a moment. We'll find that it's very similar to DNA with just a few small changes. It has the same 3 base parts, a pentose sugar, in this case ribose instead of deoxyribose. Ribose has 1 more oxygen than deoxyribose. We also see the same phosphate groups and then nitrogenous bases. Adenine, guanine, and cytosine are all the same, but on RNA, thymine is replaced with uracil. The real difference is there's a methyl group that's missing off of this one. If it were to become methylated here at this carbon, it would lose that double bond and it would become identical. It would become a thymine. But in RNA, uracil replaces thymine. Notice that RNA is single-stranded, and then the non-polar heads of each of these nucleotides would be free-floating out in space. This leads to some interesting structures as these interact with each other. We can get 3D structures out of RNA based on the order of the base pairs. Regions of charge can lead to hydrogen bonds to other regions to generate shapes like bulges, internal loops, hairpins, and actually if we have long enough hairpins we would see that that structure would form a double helical shape throughout the length of that would be similar to DNA. There are 3 different types of RNA that are important to our process moving forward. The first of the 3 is messenger RNA, or mRNA. This is the message that we just saw being made by RNA polymerase that it made off DNA. Before it can reach the next step, it needs to be modified a bit. A cap is attached to the 5' end, and at the 3' end we end up with a bunch of adenines added. These both act to protect the mRNA as it moves out of the nucleus and goes to its next destination. Before it can go, it must also undergo splicing. This is the removal of unneeded sections of RNA, called introns, and stitching together needed exons. So maybe if this section of the code is a really relatively short section of RNA altogether, but if this section was unneeded, we could splice out this section, attach those 2 pieces together, and we would end up with the completed exon, leaving out the intron unnecessary information. Notice over here we also have a triplet codon that's labeled. Excuse me again. A codon is a 3-unit coded sequence that will be important in the next step. Each 3-unit section is red. So let's go on to the next slide and see what this code might mean. Codon triplet sequences. Each codon triplet has a meaning. It calls for a specific amino acid. There are 4 possible bases for each of the 3 spots. So we end up with 4 to the 3rd, or 64 possible combinations, all being shown on this table. Ultimately, this will lead to translation, or the process of changing from nucleic acid to amino acids. Let's practice reading some. If we're looking for GCU, first we come to the left-hand side and read that first codon. So we'll find the G, and then we will find the C column. So our row is G, column C, GC, and then we need to find U. So GCU must be alanine. There we go. Next, excuse me again. Let's look for CCC. So first we'll go to that column. Here's our C. We'll then go to the row C, so CC, here we go. And let's find G. CCG must call for a proline amino acid. Proline is there, very good. Next we've got AUG, so we'll go to the column. Here's A, go to U for our column, AU, and then G. This calls for methionine. also, this is highlighted in green because that is our start codon. This is going to be that beginning sequence that will ultimately let the ribosome know when it should start creating the amino acid sequence to make the polypeptide. And finally, let's look at UAG. So if I go to, once again, column U, and then, row U, and then column A, and then go to G, we'll find UAG as a stop codon. This would code then for the end of that sequence, so that ribosome would know to release, and then we would have our completed amino acid sequence. Going back to what we said a little bit ago about different sorts of mutations that might occur, if we end up with something like a UAC that might call for a tryptophan, and it gets changed to an A or a G, then if that's in the middle of that protein sequence and now it's calling for a stop codon, half that proton will not be made. So that would cause there to be dysfunctional proteins there floating throughout the cell, and that could possibly cause some problems for the cell. Excuse me. So once again, UAG is a stop codon. Next, types of RNA. The next type of RNA is tRNA, or transfer RNA. Notice it has a few of those hairpin turns that we've talked about before. At one end, we have what's called the anticodon. It's the opposite pattern of nitrogenous bases as what we saw before on the codon on the mRNA that we had that code table for. At the opposite end, we have an amino acid. There's a location here on that RNA that the amino acid can bind to. So hopefully we can see that this piece, this tRNA, is fundamental in translating between nucleic acids and amino acids. It's literally the translator connecting the amino acid through the anticodon to the nucleic acid at the anticodon to the amino acid at the amino acid end. The final type of RNA that's important for us is the rRNA, or ribosomal RNA. There's two different halves, the large small subunit and then the small subunit, and they work together with protein polypeptides to form a ribosome. And that is ultimately the destination for the mRNA that will then lead to the next step called translation. So after transcription, the next step is translation of protein synthesis. So here we have all three RNAs coming together. The mRNA is delivered to the ribosome, which clamps around it, mRNA being clamped around by the ribosome and the ribosomal RNA and those proteins in there. Nearby floating transfer RNAs then move in place and match up their anticodons with the codons on the mRNA. This brings the amino acid heads close enough to form polypeptide bonds. More on that to come in just a moment. So ultimately, this does that step of translating RNA into amino acids or proteins. Let's zoom in and take a look at the structure of amino acids. Each one has 4 different pieces that all go around what's called the alpha carbon here in the middle. There's a hydrogen attached to the alpha carbon. There's an amino group, which has a nitrogen. There's a carboxyl group, which is this carbon with a double bonded oxygen, and then an alcohol OH group up on this side, carboxylic acid. And then we have a variable R group, which may contain nonpolar or polar groups or positively or negatively charged groups. There are 20 standard amino acids. Excuse me, and we saw the abbreviations for those called for on the table a few slides ago. Peptide bonds link amino acids together, and they're formed through a condensation reaction between the carboxylic acid group of one amino acid and then the amino acid of the subsequent one. So you'll notice this is a condensation reaction. Water is given off as this bond is formed. We lose the OH from the carboxylic acid, and then one of the hydrogens off of the amino group. So then we end up with that water. If water were to come in between these things again, we could have a hydration reaction that could occur and split this back into its original two individual amino acids. So this is a reversible reaction. The order of amino acids forms what's called the primary structure of proteins. Hydrogen bonds between hydrogens and then the oxygen and nitrogen containing R groups can form secondary structures like alpha helixes or beta sheets, also loops and turns like we saw on the mRNA earlier. These structures move into tertiary structures that are stabilized by side chains, objects like ionic bonds, disulfide bridges, and then hydrophobic interaction. In general, nonpolar groups end up facing in towards the middle, and then polar groups end up facing on the outside because they're attracted to the surrounding water, and then those hydrophobic groups are pushed away. The nonpolar hydrophobic groups are pushed towards the inside. And then finally, multiple polypeptides can link together to form the final protein complex that is the quaternary structure. So let's recap. Inside the nucleus of the cell, DNA is read and transcribed to mRNA, which is then transported outside of the nucleus after it undergoes transcriptional modification. It's delivered to the ribosome, where it's translated into amino acids using tRNA. And then finally, the resulting protein is folded into its final native or lowest energy shape, and then it performs its duty somewhere out inside the cell or outside of the cell. Thank you very much for listening to my presentation. I hope you found it informative. Here are my sources, my image sources. Although the things that I had in there were Creative Commons approved. And then my sources that I used are textbook, and then one website that I got a little bit of information about deoxyribonucleic acid, as well as one image from. Thank you very much.